Hi, my name is Trip Oliphant. This is my fourth case study in Biochemistry 1005, and today I'm going to be addressing two of the questions that are put in the assignment, or two of the case connections that are put in the assignment, and I'm going to go ahead and get started. So the first one that I chose to delve into is case number one. It's about a little girl who presented to the hospital, and then I'm kind of going to go over the deficiencies that she is experiencing. So after reading the options for the case study, I had to evaluate each option individually, and upon further investigation, I decided among the given choices that the best option would be plasma glutamine as the best diagnostic tool to use to differentiate between two disorders. And those two disorders are the urea cycle disorder and beta oxidation defect. And throughout this unit, we've learned that the amino acid glutamine is actually a non-toxic, non-essential amino acid. And while that's true, it is essential for the transfer and movement of ammonia of ammonia in molecules. It's also used for the detoxification with ammonia levels. So during this urea cycle disorder, free ammonia levels were increased as well as glutamine levels. So we compare that to glutamine levels in the beta oxidation defect. So upon further investigation of the lab results and understanding the specific enzymes that are deficient in this particular case, it's clear that this case is an example of a deficiency in the enzyme known as ornithine transcarbamylase, or OTC deficiency. This disorder is referred to as an X-linked recessive disorder and is oftentimes seen in several young males, several young males that have increased serum levels. Whereas females, like the one in this particular case, actually has a mutation on one of the X chromosomes, because we know that girls have two X chromosomes while males have one X and one Y. So whenever one X chromosome is mutated, the consequence of this includes hyperammonemia, lethargy, and apnea-like symptoms. So when a deficiency of OTC, which is what I'm going to refer to of that enzyme because it is quite a long enzyme to pronounce to kind of save time. So when a deficiency of this particular enzyme occurs, there is a molecule known as carbamyl phosphate, which begins to accumulate within the mitochondria of the cells, and it begins to leak into the cytoplasm of the cell. And when this takes place, the carbamyl phosphate is then directed to pyrimidine synthesis. So the cell has to do something with it, so it sends it to pyrimidine synthesis, which will then result in increased levels of an acid, and that acid is what we know as orotic acid. And orotic acid is transferred eventually to orotate and then excreted in the urine as urea in urine. So when diagnosing this deficiency, it's very important to observe and take note of the increased levels of orotic acid. And this is due to the fact that when orotic acid is in fact elevated, it's unlikely that the deficiency is within the carbamyl phosphatase synthetase 1, but instead it's due to a deficiency of OTC. So in summary, for the particular case, it's highly likely that due to the increased levels of orotic acid, that the enzyme that is deficient in the patient is in fact ornithine transcarbamylase, or OTC. And that would explain why the patient is undergoing the symptoms and the occurrence that she is with the certain condition. So that's it for question number one, or case number one. I hope that I explained that well enough. I'm now going to move to case number three, which is a patient who presents with soreness in one of his large toes, and it's currently been diagnosed with gout. So he's been diagnosed, the condition was known as gout, and clearly there's a significant amount of factors that play into the occurrence of this metabolic condition. So to begin, the occurrence of gout must be addressed first to determine the reasoning behind the diagnosis that the physician did. So gout is caused by a condition that we know as hyperuricemia. And hyperuricemia is caused by increased levels of uric acid within the blood. And the reason this is an issue is that uric acid begins to build up within the blood because it's not being excreted through the urine like it should be. And that usually takes place, and actually always takes place, when there's an enzyme deficiency. And we'll discuss these enzymes shortly that take place. So gout is the formation of monosodium urate crystals, and they form within the joints of chronic hyperuricemia patients. So when hyperuricemia is chronically ongoing, and the body has chronic high levels of uric acid, that uric acid is eventually transferred into what we know as monosodium crystals, and that causes the pain within the joints, known as gout. So the question to be addressed first is which option is more consistent with the patient, which option is more consistent with what the patient is actually experiencing. So I examined the urinalysis and the lab work, and between that is indicative of a classic form of overproduction of purine nucleotides. And the lab work, and therefore would be the option A. The reason for my decision is due to the fact that hyperuricemia is caused by the deficiency of enzymes used during purine degradation and salvaging. These enzymes are known as hypoxanthine guanine phosphorybosyl transferase. While in contrast, there would be an overpopulation of, so there's an overpopulation of purines right now, as we know. So they have to have the enzyme to degrade them or salvage them. And without that enzyme, they're just going to build up within the blood. And they can't be excreted into the urine because they're not being transformed. So there's an overproduction, and there's an enzyme that initiates purine synthesis known as 5-phosphorybosyl 1-pyrophosphate synthetase, which also is known as PRPP. Since there's an overproduction of this enzyme, there will be an overproduction of purines. And the reason option B would not be fully correct is because while there is a decreased salvage of purines, it is not the absolute causing factor for this condition, because there's still going to be a population of purines, but there's a deficiency in the enzyme to break it down to allow them to salvage the purines. And if we look at option C, it's likely not correct because of the high levels of urate within the blood, which cannot be excreted because it's not being transformed by the enzyme xanthine so that it can be excreted into the urine itself. So upon further investigation and talking with the patient, he obviously explains the increased consumption of sodas with high levels of fructose. With research, studies indicate the high consumption of fructose within a patient's diet can actually cause a deficiency in the process of purine salvage pathway because of the amount of ATP that's actually needed. The purine salvage pathway requires a significant amount of ATP that's needed, and the phosphorylation of fructose, which is in these drinks, is fast, but you also have to take into account the enzyme known as aldolase B, which has to be phosphorylated as well, which is actually very slow in doing so. Therefore, when this takes place, there's an accumulation of what we know as fructose 1-phosphate, and it begins to build up within the cell. And this, in return, causes an intracellular accumulation of fructose 1-phosphate and causes intracellular phosphate to decrease. So when that decreases, it in return limits the production of ATP, AMP, and ADP. And where there's a lack of ATP, the purine salvage pathway is not as efficiently working. So whereas the PRPP enzyme is still synthesizing purines and doing its job, there's beginning to be a flux of uric acid building up within the blood because it's not being salvaged or transferred back to nucleotides because of the deficiency in the enzyme known as hypoxanthine guanine phosphorybosyl transferase. So after treatment of this condition, the patient should reduce fructose intake. He should also tailor his diet to allow the purine degradation pathway to function properly. Therefore, reducing some of the fructose intake with the drinks and maybe not having as many, as well as eating things that will contribute to the purine degradation pathway. Obviously, you'd have to explain that in a different way to him, but ultimately that's what needs to happen. And this will, in return, eliminate the occurrence of hyperuricemia and therefore mitigate the symptoms of gout that the patient is actually currently experiencing. So I appreciate you taking the time to listen. I hope that this made somewhat sense in different cases. I've enjoyed the course, and I've enjoyed learning about the biosynthetic pathways in this last unit, as well as learning the biochemistry that happens within our bodies. So thank you for listening, and have a great day.